Hello students, in this module I will be talking about outbreeding devices. So look, listen and learn. They say that in most angiosperms the flowers produced are bisexual. Is that okay? You can see here two bisexual flowers. Even then what will happen is they basically prefer cross-pollination, right? Yes, so what you saw here was cross-pollination, right? And these plants then develop something called as outbreeding devices. Or you can also say they are basically methods to avoid self-pollination, right? So let us see what are some of the outbreeding devices you will see in angiosperms. What you see behind me is a beautiful case of outbreeding or cross-pollination. There are four basic methods that have been seen in outbreeding devices. Number one, dichogamy. Number two, heterostyly. Number three, self-sterility. And number four is unisexuality. Let us understand what these four are individually. So first let us see what is dichogamy. Now dichogamy basically is a phenomenon or a condition where stamens and carpel do not mature simultaneously, right? Let us see what this really means. Now what you see here is the beautiful bisexual flowers. What you see here is the anther basically is very fertile. It is developing pollen and the pollen is ready to go and fall on the stigma, right? What you're seeing here is the pollen is trying to get to the stigma, but the stigma is not receptive yet because the gynoecium has not yet developed, right? So what you're seeing in this situation is that the stigma is not receptive even though the anther is producing a lot of pollen grains, right? So this situation is called as protoandry. Protoandry is basically observed in sunflower, and protoandry is where androecium develops before gynoecium, right? There is one more situation in dichogamy. Let us see that. Now once again you are seeing another bisexual flower. Here the stigma is receptive, but look at those anthers, they're empty, which means they are neither producing nor releasing any pollen grains. So this situation is called as protogyny. Protogyny is observed in a plant of Michelia. Look at that beautiful flower. What you see here in protogyny is the gynoecium develops before the androecium. So in both these situations, they would actually choose cross-pollination over self-pollination, hence outbreeding device, right? The next situation is called as heterostyly. The name says it. When plants produce two types of flowers with different length of styles, the phenomena is called as heterostyly. Let us look at the picture here. So in this first plant, what you see is a short style. In the second plant or the second flower, what you see is a long style, right? Now in both cases, what you see, there's a barrier between the androecium and the gynoecium because of the different length of the style. And this situation is called as heterostyly. Heterostyly has been observed in primrose or primrose, right? If you see that first flower there, see that style is very short. So the stigma is at the lower level and the anthers are way up on the top and they cannot exactly send the pollen down there. In the second situation, you see a flower where there's a long style, right? And the anthers are down here, right? And what happens is they cannot throw the pollen all up there. So in both situations, because of heterostyly, the only choice what remains for them is cross-pollination, and they avoid self-pollination. The third situation is called as self-sterility. Let's understand what is self-sterility. Now look at this bisexual flower here. Now when the time is right for pollination or for self-pollination, what it has to do is basically has to transfer the pollen from the anther to the stigma of the same flower. And when the conditions are right, then what will happen? That the pollen will germinate and a pollen tube will be produced and fertilization will be achieved. This is a normal situation. But what happens in self-sterility? In self-sterility, what you see is even though the pollen is transferred onto the stigma, but the stigma is not letting the pollen germinate. Why? Because the two are incompatible. So they say when the pollen germination on stigma of the same flower is inhibited, it is called as self-sterility. It is also called as self-incompatibility. And you observe this phenomena very much in orchids, right? And also remember, self-sterility actually has a genetic mechanism associated with it. Okay? So that is self-sterility. So in this module, I've talked about outbreeding devices. In the next module, I will specifically talk about unisexuality as an outbreeding device. Till then, thank you and take care.